Okay, so hello and welcome everyone, and thank you all for joining today's webinar, which is centered around Randox's signature Biochip Array Technology. My name's Claudia. I'm a senior sales executive here at Randox based in Melbourne. And joining me today is Jamie Rankin, our National Sales Manager of Randox, who has kindly stepped in Damien's absence today. Over the next 30 minutes, we aim to share the exciting potential of biochip array technology to improve clinical diagnostics and enhance your research. Now, there's a broad array of applications for biochip array technology, both for research and for clinical use. So we hope that each of you can walk away today with some interest and excitement for our multiplex testing technology, regardless of what type of clinical or research area you're from. If you have any questions, please feel free to pop them into the chat box, and hopefully we can get to these at the end of the session. Or as always, you're very welcome to reach out to your local representative from Australia, New Zealand, or Japan for a private discussion. So we realise some of you online today are very familiar with Randox, but others may not be. Randox was founded in 1982 in the UK, and back then we primarily manufactured clinical chemistry reagents and calibrated materials. We have since grown and expanded our range to now provide different areas such as chemistry analyzers, toxicology, molecular food diagnostics, our external testing services, including RICUS, our immunoassay analyzers, which we'll talk about today, and most recently our Randox Health clinics. So we've now become quite a global market leader in the IVD industry. Some of the achievements we've had over the years now, we manufacture 4.5 billion assays every year. We're the third largest manufacturer of clinical chemistry reagents and the third largest manufacturer of QCs and calibrators. We have over 3,300 staff and a global presence across 145 countries. In Australia, we operate as a central hub with our own office, our warehouse, and our own administration and logistics staffing. The products we supply go to over 100,000 laboratories globally, and we estimate that because of this, over 400 million patients are diagnosed with some Randox technology every year. We commit 16% of turnover into R&D. We have, as I mentioned, the local support across Australia and Pacific because we are a local hub of Randox. We offer our own local technical support, installation and engineer support, us, the dedicated team of sales professionals, and we also utilise 24-hour support through our colleagues in the UK. A big focus of our R&D work has and will always continue to be our Randox patented biochip array technology. So this was created first in 1992 and it's had over £250 million investment to date. Basically, biochip array technology, it's a chip-based immunoassay or molecular assay to detect multiple biomarkers from one single sample. And the technology works by combining a panel of related tests on a single biochip with a single set of reagents. And the aim is to provide as much information as possible, faster and more efficiently than traditional methods, such as ELISAs. This kind of highly sensitive and unique multiplexing unlocks so much more potential for diagnostic and research than singular tests alone. This ability to detect many analytes from one small sample gives you that ability to detect biomarkers in conjunction rather than focusing on just one or a particular subset of biomarkers due to perhaps your time or cost or resource limitations. So some examples here are perhaps where we've always analysed interleukin-6 as a cytokine marker. We can now start to analyse a cytokine storm through a multiplex panel. or where we've only focused on one biomarker and how this may be reacting to our novel treatment, we can now assess the response of a wide range of biomarkers to our particular treatment. What is a biochip exactly? So a biochip is a 9 by 9 millimeter ceramic chip, which acts as the reaction vessel. And this replaces multiple cuvettes or wells in your 96 well plate. Each biochip is prefabricated with an array of discrete test regions, and these discrete test regions contain immobilized antibodies or complementary oligonucleotide probes, depending on our particular array of interest. And it's based on ELISA principles, so the reaction relies on the binding of each analyte from the sample to each discrete testing region. So you can see this image here, and it shows 9 of our white ceramic biochips inside the biochip carrier. And each biochip is held down in the carrier by the prongs, and this ensures that the biochip orientation stays the same, and then it minimizes the risk of the user coming into contact with the sensitive biochip surface. You can see here pointing off, I have a graphic of the discrete test regions. Now you can't actually see the discrete test regions if you hold up a small biochip, but I've popped these here just for our reference. So each discrete test region will hold an immobilized antibody or oligonucleotide probes, and this acts as the site that detects that particular biomarker when you put one sample into the well. So now I know what you're probably thinking. There's only 9 wells in one biochip carrier, and what if I want to analyze more than 9 samples at any one time? Well, we supply a biochip carrier tray, which fits 6 biochip carriers inside the tray, and you can perform basically all the steps with the entire tray of 54 samples. So you could either go for your 9 samples or duplicates of 9, or you could work with a tray of all 54 samples. And these are the steps that you would take in working with the biochip. So you would add your diluent, your sample calibrator or QC, then you would add your reagents. You would perform one or a few incubation steps depending on the particular array. And the incubation steps are performed in this incubator here called a thermoshaker, which Randox also provides. You would then wash the biochips, and this taps away any unbound conjugate. And then you'll be ready to add the signal reagent for each biochip. And the signal reagent we use is luminol peroxidase. And this signal reagent creates light output at each discrete testing region, which has had hybridization or in some cases competition by the biomarker. And the label, the detection enzyme we use is horseradish peroxidase. The light signal coming from each discrete testing region is then captured by a charge-coupled camera using chemiluminescence. And this camera is held up here within our proprietary analyzer called the Evidence Series Analyzers. This one in particular is called the Evidence Investigator. So you would, after you add your signal reagent, you would insert your biochip carrier, all your 9 samples into the instrument, and then in 2 minutes, the camera takes its images and quantifies the amount of light coming from each discrete testing region in relative light units. These are then compared with a calibration curve, and then it translates your results into units. So I mentioned there that the addition of the signal reagent causes a light output where a biomarker is present in some cases, and in some cases where the biomarker is not present. And this is because Biochip Array Technology uses 2 different ELISA type principles, which I'm sure you're quite familiar with. The first of which is a sandwich immunoassay principle. This means that the more analyte in a sample, the more conjugate will bind to form a sandwich complex, and that the biomarker concentration is directly proportional to the light signal. Some other arrays use competitive immunoassay principles, which means that the more analyte present in the sample, the less labelled conjugate will bind to the reaction site because there's a competitive reaction happening, and that the biomarker concentration is then inversely related to the amount of light generated. Some other arrays we have created more recently are the molecular-based arrays, where we're now hybridizing not an antibody at a particular discrete testing region, but an oligonucleotide probe. So these sequences are hybridized on different discrete testing regions depending on our panels, and this allows for pathogen detection, for mutation detection, or for genotyping of samples at single nucleotide polymorphism levels. So Jamie will go through our range of arrays and their applications, but now I wanted to touch on the evidence range of instruments. So as I mentioned, all of our biochips must be imaged and processed by one of the Randox Evidence Series instruments. And these instruments are tailor-made in-house to fit the biochip carriers and image these at the highest accuracy. So we'll start with this particular instrument, and this is called the Evidence Investigator. And it's Randox's most well-known biochip array technology analyzer. It is the semi-automated analyzer, so it does require your manual pipetting of your sample, calibrated control, reagent, and your manual wash steps. Therefore, it is most suitable for research laboratories or clinical laboratories, but yes, it certainly does require a laboratory environment. But then, yes, all the imaging, analysis, and reporting is automated. There is a walkaway time during incubation, which can vary between the different panels. It can be, some panels have an incubation time of 1 or 2 hours, and some have overnight incubation required. The analyzer is supplied in a package together with the handling tray, so you can work on 54 samples at once, supplied with the thermoshaker for your incubation steps, with the barcode scanner, and that allows for your reagent batch and calibrated batch information loading, as well as you can scan some sample IDs if you do that within your laboratory. And then the PC as well. So the PC is required just next to the analyzer to set up your work list, to view your results, and then there's the ability to export your results as you please. The analyzer has quite a small footprint, so it stands up just on a bench at about 75 centimetres tall, and it's only 24 kilos, so quite suitable for any laboratory bench. And because there is only one moving part to the instrument, there's minimal maintenance and servicing required. The next member of the evidence family is the Evidence Plus. So this is Randox's fully automated biochip analyzer. And Randox has quite geniusly taken the entire workflow of the evidence investigator and automated it. So now all you need to do is load your samples onto a sample carousel, which sits in here on the left. Load some fresh biochip carriers into this stacker, and then this entire stack is inserted into the loading port. You'll then specify which test to run with which sample in your carousel, and can sit back while the analyzer does the rest. So inside, it will do all of your steps similar to the evidence investigator, but automatically. So the addition of your diluent, your sample reagent conjugate, incubation steps, and imaging steps will all be done on board with walkaway time. The reagents are all stored in bottles in this bottom section, and then are just replaced whenever necessary. So we typically see this instrument in larger clinical laboratories where the department needs a higher throughput, faster, perhaps more automated than what the evidence investigator could offer, such as toxicology departments. And Jamie will touch on this a bit later, that most of the toxicology arrays that we provide are suited towards the Evidence Plus instrument. It can achieve 120 samples per hour, with your first result available in as little as 45 minutes. And because it has automated pipetting, the sample volume actually reduces quite low to 6 nanograms per litre. The evidence multistat is the third instrument we'll touch on today. So this is our fully automated point of care biochip analyzer. So Randox has taken the concepts and workflow from the evidence investigator and taken the automation aspects from the evidence plus and asked, well, how can we transform this into a point of care solution so that clinicians can get these results outside of a laboratory setting? And we've done this through the use of a pre-filled cartridge. This is the cartridge here, and it includes 2 biochips. So it looks quite different to the biochip carrier we saw for the previous 2 instruments. This includes just 2 biochips. Inside this middle section, we'll see all of your reagents, assay diluent, conjugate, and signal reagent. And these 2 wells are for your sample addition. So you would pipette your samples into the sample slots here. All of your reagents in the middle here are actually covered by a foil seal. Then you would place your cartridge straight into the multistat analyzer, and on board the analyzer it has pipettes that would pierce the foil packaging in the cartridge and pipette your necessary reagents into the biochip just as we would manually when it's needed. It requires special cartridges. So currently we only manufacture arrays for certain disease states and areas of the laboratory. So at the moment, we're focusing on point of care toxicology. So you're mainly your common drugs of abuse panels and how we can transform these into a point of care test using Biochip Array Technology. But we actually have a few panels upcoming for multistat, which includes stroke, ARDS, and cytokines. So Jamie will take you through the different biochip arrays, but I did want to mention here that the arrays, they're all prefabricated, and this means that you can purchase these off the shelf whenever you need to. Each panel is carefully considered by our expert R&D team, and the biomarkers that we create and the antibodies and aptamers that we hybridize to the discrete testing regions, they're based on latest research in that particular field. All of our antibodies and aptamers too, they're manufactured internally by Randox in the UK to our strict quality guidelines so that we can have a high standard of testing with CVs typically less than 10%. Okay, so now, Jamie will take you through the range of biochip panels available.
Okay, hello everyone. Thank you for joining our webinar today. As you've seen, Claudia has really took you through what the Biochip Array technology is and how the arrays are designed and developed. This part of the webinar, I am going to take you through the various different arrays that Randox can offer. As we do have time constraints today, we wouldn't be able to go through every single array that Randox offers, so we'll go through the most common arrays. We have a range of immunoassay arrays, going to our molecular-based arrays, and then we offer drugs of abuse arrays. Okay, so the first array that we would like to introduce is the cytokine and growth factor array. So cytokines and growth factors have been linked to many different disease states, such as autoimmune, allergy, vascular disease, and cancers. Randox offer a comprehensive menu of 26 cytokines, cytokine receptors, and growth factors across 4 multi-analyte arrays. So the first cytokine array that we'd like to discuss is the cytokine array 1. So cytokine and growth factor array 1 is the most commonly requested cytokine panel here in Australia and around the world. The biochip contains 12 of the most common cytokines and growth factors and is suitable for either clinical use, as it's currently CE marked, or for a research setting. This panel has had many reviews and publications around the world, with several tertiary hospitals, including France, the Netherlands, and Vietnam, using this current panel routinely on their evidence investigator. So we just wanted to give you an example of why it would be beneficial to run the cytokine 1 array on one of the Randox Biochip Array instrumentation. So if a clinician or a user was to run the 12 different biomarkers that we listed in the previous slide, they would simply have to do 3 96 well plates. They would have to purchase 12 ELISA kits. This would require 2.4 milliliters of sample, which is 100 microliters per test. And it would take many, many hours to run all of these biomarkers. How would that look on the biochip array, cytokine 1? Well, simply one carrier, 9 biochips, so you can run 9 samples from one biochip. This would only require 100 microliters of sample, which is 8.3 microliters per test, and you would get your cytokine results in as little as 3 hours. So this is quite significant, this difference in being able to save time and costs. So in addition to the cytokine 1 12-plex array, we do offer 3 additional cytokine arrays for your more unique biomarkers. They're currently for research use only. So the next, I would like to introduce the Randox Alzheimer's disease array. So this is a brand new addition to our range of biochip panels and has recently been CE marked for diagnostic use. As we know through current research, Alzheimer's disease is 14.9 times more likely in individuals with the ApoE4 isoform. Randox developed an array to detect the proportion of ApoE4 compared to the total ApoE directly from a plasma sample. You can then get your results by running this array in as little as 3 hours. So this array offers a rapid and highly sensitive blood test facilitating direct ApoE4 genotyping without the need for molecular genotyping and has shown to have 100% concordance. This new Alzheimer's disease array is available on both our semi-automated Biochip Array Technology, the evidence investigator, and also our floor standing Biochip Array Technology. So meeting the needs of all users. So thyroid arrays. Randox currently offer 2 CE marked diagnostic thyroid arrays. These arrays allow for the numerous detection of thyroid hormones from a single patient sample. This allows for the screening and monitoring of hyperthyroidism and hypothyroidism. So you can see here on the screen the 2 arrays that Randox currently offer for thyroid. These both can be run again on our benchtop semi-automated evidence investigator, but can also be run on our largest Evidence Plus machine. So Randox Gastrointestinal Biochip Array offers a comprehensive and non-invasive testing method to assess gastric health and individuals that are at risk of developing gastric cancer. It is capable of detecting 3 stomach targets in conjunction with Helicobacter pylori array for efficient screening of patients at risk. There are currently 2 arrays available for this category. The first one being your Helicobacter pylori array, which quantitatively measures Helicobacter pylori IgG only. Helicobacter pylori infections are very common, with as much as 50% of the world's population having been infected. In addition to this, we also offer the GastroPanel array, which contains 3 unique biomarkers which implicate in gastric cancer risk. Both these gastrointestinal biochip arrays are available for the Biochip semi-automated benchtop analyzer. So moving on, we've looked at a few of our immunoassay arrays. We now want to discuss some of our more common molecular arrays. So this is our molecular STI array. As Claudia mentioned, these arrays are oligonucleotide probes on the DTRs instead of antibodies. This allows the biochip to detect genetic sequence of variant pathogens in biological samples. The Randox STI biochip array is the most broad STI microarray on the market today. It combines 10 bacterial, viral, and protozoans and pathogens in one biochip. It can provide results of infection and co-infection in as little as 6 hours. In addition to our STI array, we also offer the molecular array for respiratory pathogen detection. So another molecular array which has been featured a lot, especially in recent times of respiratory pathogen awareness, is the respiratory pathogen biochip. It is a 22-plex array combining the most common viral and bacterial pathogens. So rather than a clinician prescribing a general course of antibiotics, we can detect the exact pathogen causing the infection to specialise the patient treatment plan. Using this array on the evidence investigator can provide results in as little as 6 hours. This is currently CE marked for diagnostic use. So our molecular arrays are not only for pathogen detection, but for detection of specific genetic sequences that we know are implicated in certain conditions. Our colorectal cancer array, for example, detects mutation status of 3 common genes we know to be targets of colorectal cancer: KRAS, BRAF, PIK3CA. So unlike full sequencing methods, this biochip panel allows for the rapid mutation detection at a lower price point. Again, this array is CE marked for diagnostic use. So going on from our molecular arrays, we just wanted to touch on drugs of abuse arrays. So Randox currently offer drugs of abuse arrays for 3 different instrumentation, really covering a wide range of toxicology needs. We offer a point of care machine, we offer a semi-automated benchtop machine, and then we offer that large floor standing machine for your toxicology testing. So these toxic drugs of abuse arrays are some of the most common arrays we've seen in clinical toxicology and forensic settings around the world. The panels we design are all suitable for primary screening of drugs and drug metabolites in samples. Randox offer over 21 different drugs of abuse arrays covering over 600 different drugs. We couldn't go into all of them today, we just wouldn't have time, but to give you an idea, we cover the most common drugs of abuse in the market and also offer new psychoactive substances. The toxicology panels that are offered for the Randox Biochip Array machines are some of the largest offered in the world. Up to 44 analytes can be detected per biochip. The toxicology panels are specifically manufactured for different matrices. So there are panels designed for blood, urine, oral fluid, and hair, just to give a few. Our toxicology drugs of abuse arrays are suitable for forensic and clinical toxicology, workplace drug testing, rehabilitation, and racing. So we couldn't go into all of the toxicology arrays that we offer today because we have over 21, but one of the ones that we did want to discuss is Randox recently introduced the Multiplex. So this is a semi-custom way of designing your own drugs of abuse panel for the multi-stat. So you can see here on the screen, this is 29 different drugs of abuse analytes. What can happen here is the user, when they want to run this drugs of abuse array on the multi-stat machine, can choose to run all 29 analytes, or they can choose to run 10 analytes and view the results for 10. What's really great about the Multiplex array is that it's also customizable in the fact that you can choose the cutoffs that are specific to your market and that are relevant to your testing. So lastly, we wanted to touch on an exciting upcoming panel. The Randox Stroke Array will be a rapid and highly sensitive blood test to complement and enhance existing CT scanning technology for stroke diagnosis. This panel will be available on the multi-stat or point of care instrument, making it very suitable for emergency settings. The biomarkers that have been selected and designed, they are designed to differentiate and classify patients with either ischemic or hemorrhagic stroke. So this is a really exciting array that is coming up that will be available for the multi-stat, the stroke array. It's really relevant for those emergency settings because it provides results in as little as 30 minutes. So we all know that when we do webinars, even though we try to explain the best we can, the products we offer, the technology we have, we all know that there is many questions. So we just wanted to put up some of the most common questions we get asked when we discuss the biochip. So how many biochips are included in the kit? Well, typically there's 54 biochips included in a kit. However, your Evidence Plus, so the larger floor standing machine, their kits are supplied with more biochips because these instruments are gauged more towards your higher throughput laboratories. So which components are supplied in the kit? Well, basically everything you need for that particular array, except in some cases, for example, your cytokines, you would need to purchase the quality control material along with the cytokine array. So in the kit, you would get adequate reagent, conjugate, luminol, etc., along with the 54 biochips. Do you need to run all 9 biochips in a carrier at the same time? This is a question we get asked all the time. What if I don't have 9 samples yet? Well, that's no problem. You actually can run less samples on the biochip. So you can run a minimum of 3 biochips, and then you can save the remaining biochips for another time. They just need to be popped out. So how often is calibration? Well, that really depends on the array that's run. So if there was arrays of interest during this presentation, please do reach out or drop it in the chat and we can send you through the inserts so that you can have a look at the calibration. So we just wanted to really thank you for all joining the webinar today. Apologies that it's run a few minutes over time. We really do appreciate everybody taking time out of their day. If there is any questions, feel free to drop them in the chat box and we could answer them now, or feel free to reach out to your local representative or any of us and we can help answer those questions. Thank you very much for attending today's session.